Thanks for the introduction, Tom, and thank you all for coming, and thanks for the organising committee for asking me to speak to you tonight about an aspect of work that is relevant to all of us. And as you know, the topic for this series is climate change, and I've been preceded by a number of colleagues who have addressed this from a climate point of view, from a point of view of geography and a point of view of vertebrate animals, and I'm going to be talking about pest insects. And I'm going to be honest straight away and say that this is not my area of direct expertise, although I'm very well versed in genetics and, as Tom said, looking at issues of mosquitoes and mosquito-borne disease. And so this impacts on a lot of what we do and a lot of what's going to go on in the next 10, 20, 30 years as we address issues of climate change. But I say that in the context of this talk, in that I'm going to be referring to a lot of work by colleagues around the world. And it just is a matter of principle that when I do this, I'm going to be referring to their work and show references at the bottom, just so you know that we're talking about work that's being done by a number of people around the world. And I'm just going to try and synthesize that for the next 50 minutes or so, and then towards the end actually talk a little about what we do in the lab and how that's relevant to the general thesis that I'm trying to propose. So the topics for discussion tonight, what I'm going to be trying to cover is that I'm going to go through the evidence for climate change. I'm not going to spend too much time on this because certainly the first two speakers addressed this, and I'm just going to use a few of the types of slides that they used just to refresh our memory about what we're dealing with. I want to give a little primer on the importance of insects for human welfare and well-being, both beneficial and pest. We experience them more as pests, but I want to illustrate to you how abundant they are and how dependent we really are on them for things that we never even really think about. I want to very briefly talk about the effects of temperature on insects. This will be very, very brief, and the bottom line virtually is that temperature does have a profound effect on the physiology and development and behaviour. And then I'm going to go through some fairly detailed data showing what some predictions for the distribution of malaria may be in the future, and illustrate to you, I hope, how difficult this is to measure and how controversial this topic can be. And I really want to bring to you the idea that what's important when we deal with all these things is the question of measurement and how precise that measurement needs to be and how difficult that can be sometimes. I'll then talk about an example more recently from the 2006 El Nino event in Southern California and the impact that had on some diseases around the world. And then in the final part of the talk, I'm going to be talking about genetic variation and how this is a bit of a black box at the moment, but the technology that's being developed around the world and here at UC Riverside is going to help us in terms of developing predictions for how many insects will respond to climate change in the next 10, 20, 30 years. This is an interesting quote from a very famous textbook in entomology written by Howard Evans, who was at Colorado State, at least when he wrote this textbook, and it's called Insect Biology, a Textbook of Entomology. And my edition is the 1984 edition, and one of my favourite quotes in terms of trying to marry genetics with entomology is that insect-human relationships are a contest between human insight and the genetic capacities of insects. So when we talk about insects competing with us for resources, and I'm talking primarily now about pests, so we're talking about mosquitoes that transmit pathogens like the Plasmodium that causes malaria or viruses that cause West Nile. When we are trying to come up with strategies to counter the effect of those insects, ultimately we're addressing the genetic variability of those insects. Even if we don't know it at the time, even if we have at that time no way to measure that, ultimately it's the genetic variation in the insects that we're dealing with. And one of the most compelling reasons for doing entomology at the moment and in the future is, as I hope you'll see, that in the years ahead we now have the genetic technology that's catching up to the complexity of insects and the diversity of insects, and so enables us for the first time to address variation directly. And in relation to climate change, which is the subject of these lectures. Okay, so a quick summary of where we've been. This is where we live. As far as we can tell, everywhere we look, we have found no signs of life anywhere else, although we're finding tantalising signs of water elsewhere. We're surrounded by a universe with billions of stars and most likely billions of planets, but to the best of our knowledge, this is the only place where there is life. But probability tells us that in the centuries and eons ahead, most likely we will find life elsewhere in the universe, either directly or by monitoring emissions that we get from there. And one of the defining aspects of our species is that we can contemplate these issues. We can talk about why we're here, how unique that is, and what our stewardship of the planet has been like. Another defining aspect of our species, and there are many, is this graph. And this is an old friend of us if we've been attending these lectures. And there are three things that at least stand out to me when I look at this graph. And Dr. Kennedy presented this some 6 weeks ago. And what we're talking about here is this final little peak. Well, it's actually a large peak. And this is the concentration of carbon dioxide in the atmosphere measured in parts per million. And this is once again, I'm not using my own data. This is coming from NASA. And as it says here, for over 650,000 years, atmospheric carbon dioxide has never been above this line until now. And here is 1950, which is at the point 0 here. And you can see that why we're having this lecture series and why climate change is such a current topic is this actual peak. And we have to own these data. This is bad news. This is actually pointing us to a possibility that we're heading to a place we don't want to be. And we have to own these data. So I was born in 1958. And I'm certainly not responsible for everything past 1958. But I am certainly a tiny part of the issue in that the lifestyle that I choose or did not choose, I was just born into it, directly leads to this sort of thing. And so we have to actually think about how we as individuals are causing this and then trying to change it. So it's like bad medical data. You can deny it or you can accept it and say, what am I going to do about it? But in this case, it's a lot harder problem to overcome because unlike medical data, which gets down to you, this gets down to all of us. And not all of us may agree. The other two things which actually strike me here is that I find it remarkable that we have the ingenuity to actually measure carbon dioxide concentrations going way back, in this case, 400,000 years. That's a tribute to the ingenuity of the scientists who can actually come up with strategies to determine carbon dioxide concentrations and temperatures in eons before we were even around. And the other thing that strikes me about these data is the oscillations. And we've heard from previous speakers that many of these are due to the natural course of orbit around the sun over the years. And what I've also drawn up here are some events that might be of interest to us to put this into context. Homo sapiens was thought to emerge in Eastern Africa at some time between 250,000 and 160,000 years ago. So here's this red bar. Here is the human migration from Asia to North Africa, or North America, I should say. Here's the time when domesticated dogs, or dogs that would become domesticated dogs, split from the wolves. And then in this little bar up here, which sadly is trying to represent accurately, but I think I have failed, 10,000 or 12,000 years, this is the course of time when we've actually been able to develop agriculture. And as has been pointed out by many authors, this is consistent, our ability to develop agriculture is being consistent or correlates with the time of stability in the climate until quite recently, and stability in sea levels. Here are some more data from NASA, simply showing, going back to 1880, the increase in temperatures from 1880 to 2000. Once again, it's remarkable that we can measure these things, and these are done relative to the temperatures around about 1950 to 1960. So you can see that this is a constant rise. Oops. And then here's more data, initially from tidal charts, and then more recently from satellite imagery, showing that indeed there is an increase in sea level, going back to 1870 if we look at tidal charts, and then more recently with satellite imagery, and here's the rate of change. So all these parameters that has been discussed about as being evidence for climate change are real, and I was just touching on them there. And it all gets down to our ability to measure. And as has been shown before, we understand the ultimate cause of, the source of heating on Earth, even without the presence of humans. We have solar radiation coming into the atmosphere, and most of the outgoing heat is absorbed by greenhouse gas molecules and re-emitted in all directions, warming the surface of the Earth. So this occurs naturally to some extent with a normal atmosphere, but if we're pouring these greenhouse gases into the atmosphere, we're simply making it worse. And then one of the most dramatic examples, which really brought this home to all of us, especially those of us who lived in the southern hemisphere, occurred in 1986 when, once again, satellite imagery and measurements illustrated to us that ozone was being eroded around the South Pole, and that led to this ozone hole, and actually led to a galvanisation of global opinion to do something about this. And I hope maybe you can see this. This is data from NASA once again. This is the ozone hole from the last southern spring in September 17th, and this was the ozone hole at its maximum. Here's Antarctica, here's South America, and New Zealand. I think Dunedin is just around over here somewhere. You can't really see it. But this is still real. It's not growing as large as it used to, but it's still real. And I can tell you that for some years when we lived in Australia in the 1990s, there were times when the very edges of this ebbed up to southeastern Australia, and you would get warnings in the press about going out and just being aware that you may get sunburned a little easier than what was you would normally get sunburnt in the summer. Okay, so much for the reminder about global warming. Now let's talk very briefly about the abundance of insects. We see ourselves as the ruler of the planet, and there's lots of reasons to believe that we really are. But if we talk about the sheer numbers of organisms on Earth, bacteria, plants, animals, it's the insects who rule. So what this fairly boring pie chart is showing you is the number of characterized species. So we're one species, right? Homo sapiens. And we're mammals. And if we look where the mammals are in this pie chart of the numbers of species, I'm not talking about absolute numbers of individuals, just numbers of species. And there's what, 6 billion of us. But maybe 6 billion of us, but we're counted as one species. And it's up here. These are the mammals. And if we look at all the characterized species on Earth, then the mammals make up less than 1% in terms of the numbers of species identified, and we're one of those 4,170. Okay? There are 751,000 characterized species of insects. 52% of characterized species on Earth, prokaryotes, eukaryotes, are insects. And then if we start talking about absolute numbers, then I have no idea, but I bet it's more than 6 billion. Okay? Here are non-insect arthropods like ticks, spiders, and that's 132,000, 9%. And here are the flowering plants. So insects are the most dominant animals, the most dominant creatures on Earth when we talk about absolute numbers. And that means that they're contributing massively to the biomass. So Howard Evans, once again, made a nice calculation in his textbook and said, if we estimated all the insects on Earth and weighed them per acre in the United States, we'd have about a biomass of about 400 pounds of insects per acre in the United States. And if we just looked at humans, that weight would be about 14 pounds. So I want you to think about that term in just terms of sheer biomass and weight. Now here's a more interesting depiction of that pie chart, and this is from a wonderful textbook of entomology by Penny Gullen and Peter Cranston of UC Davis, who formerly were at CSIRO in Canberra at the same time as we were there. And this is actually showing all the creatures on Earth, and instead of slices of a pie, the size of these creatures are shown in proportion to the numbers of characterized species. And we can thank heavens that this is not how that disproportion of numbers manifests itself. And I should also point out, if you ever ask an entomologist why you study entomology, then actually this slide answers it all. Because here's a blowfly, and it's enormous in this depiction. Here are the mammals, so it's an antelope or a deer. I'm not a good enough taxonomist to know the difference. It's tiny though. And remember, there was something like 4,200 species of mammals, and so we're one of those. So if we were going to draw a little picture of a person or a man or a woman here, it would be tiny, and if we said how many people are actually entomologists, it would probably be a microscopic dot relative to the number of organisms, number of insects that are out there. So insects are abundant, and the majority of them are beneficial and do not harm us at all. A small minority are pests, and they compete with us for food resources, and some of them make us sick through vectoring or carrying disease. One of the most favourite beneficial insects are bees, whether they're honeybees or wild bees. And this was an interesting article from the New York Times back in March 25th in response to a phenomenon called colony death syndrome. But there was just some nice data that I just took from the New York Times, and it pointed out that a third of agriculture relies to some extent on pollination caused by bees, but bear in mind that less than 10% of the 100 most productive crops depend on pollination for propagation of the plants. And elimination of all these bees, honeybees or wild bees, most of the time this has been done by wild bees, would probably reduce global food production by 6%, which is not a huge amount, but it is still a significant amount. And then over the past 50 years, the amount of agriculture that has depended on pollination has increased in the order of 300%. And that's an important point, because the types of crops that require pollination in order to reproduce typically need large areas of land in order to grow. We're talking things like apple trees and avocado trees, not crops like wheat, barley, which can be intensively cultivated in small areas. So let's pretend in a worst case scenario that all the bees go away and here's an impact. But this simply illustrates that honeybees or bees are important. They're beneficial insects. In fact, most insects are beneficial. They eliminate waste. They eliminate leaf litter. They eliminate animal waste. Dung beetles in Australia eliminate cattle dung. And it's remarkable to watch how efficiently they work. And many insects, of course, form parts of food chains, which we all depend on at some stage in that chain. So in this talk, so there's our introduction. In this talk, I want to talk about measurement and prediction. And I've drawn 3 black boxes. And the first black box is climate. And this is in the physical sciences, of which I'm not an expert. A second black box talks about dynamics of disease transmission that is vectored by insects. It can be a mosquito that transmits a virus like West Nile virus, or a protozoan like Plasmodium falciparum, which is one of the causative agents of malaria. Or it could be an aphid, a pest of agriculture, which transmits viruses. But there's a dynamic of transmission, the interaction between the pathogen, the germ and the insect, and whether or not that insect is going to be in a position to actually transmit that pathogen. And we're going to go into some detail about that when we talk about malaria. And then finally, insect genomics. All right, in other words, what's the genetic constitution of these pest insects? And what I want to illustrate to you by the end of the talk is that, in fact, this black box is sort of vanishing through the efforts of our climatologist colleagues and our who have actually illustrated to us quite graphically what the problem is through measurement. All those slides I showed you at the start come through being able to measure tiny amounts of molecules and do all the proper controls and come to conclusions which really have an impact upon us. And I'm going to say that we still have 2 black boxes here, and I'm going to illustrate to you hopefully that even those are starting to erode away. But we're still far way behind our friends in the climate world. Very quickly, what are the factors affecting insect distribution? If we're going to be talking about the impact of climate change on insects, we can worry about whether insects are going to be eliminated, and hopefully I've convinced you that because most of them are beneficial, that would be a bad thing. But we're going to be primarily talking about pests, and because of my expertise in mosquitoes, I'm going to be talking about insects or mosquitoes that transmit malaria. Because one thing you'll hear about and one thing you read about is, oh my gosh, there's malaria in the world, and we'll briefly talk about that. It's going to spread. Things are going to get warmer and it's going to spread. And what you need to know, what we all need to know is, well, is that likely or is it not? Based on all the scenarios we've seen with climate change. So what are the basic factors affecting insect distributions? Lack of opportunity to disperse. There is no insect species on this planet that normally, naturally, has a global distribution. Those, like houseflies, like Drosophila, that do have a global distribution have so because of our activities. And my favourite example is the Australian sheep blowfly, which you would think is Australian, but it comes from South Africa. And it came to Australia because during the colonisation of Australia, English settlers took on livestock in Cape Town and then transported them to Sydney and to Hobart. And of course, they weren't entomologists. And some days later down the track, when these horrible blowflies were in Australia, of course, they had to be Australian blowflies, but they're not. The lack of suitable habitat or host plant for the animal. So clearly, if an insect that feeds on plants, if those crops are not there, then it's not going to have the environment in order to disperse. And then temperature and moisture. And clearly the last two impact us the more when we talk about climate change. And then what are the attributes of a pest species? And there could be many of these, but I'm basically only going to be talking about 4 very briefly. High reproductive rate, that is the ability to leave lots of offspring and to breed quickly. The ability to disperse rapidly. Their small size, so think mosquitoes. And an ability to adapt. And we'll talk about the genetic variation needed to successful adaptation later on. But those of you who sometimes would leave kids' swimming pools in your backyards, as I did once, and not tend to them for a week, and I probably had to hand in my entomology license if such a thing existed, can be amazed when you come back a week later in the middle of summer and find out how many mosquito larvae can be in that pool, simply because these mosquitoes are pests. Okay, we're going to be talking about malaria, and I want to go into detail. And as I said, one of the reasons this comes up as one of the exemplars of being concerned about the possible effects of climate change on mosquitoes is, well, are we going to widen the areas of malaria? Because when we think of malaria, we think of that disease being in the tropics and the subtropics. And if the world is warming up and that there's greater rates of change in temperate regions rather than tropical regions, are we going to have malaria moving north and south depending on what side of the equator you're on? So here's the current global distribution of malaria, shown in basically varying shades of green. And it's in Africa, South Asia, Southeast Asia, and Central and South America. It exacts about 1 million deaths per year. Whilst I give this talk, 100 people will die. There's basically 300 to 500 million cases per year. And in many of these countries where it's an endemic, it's a driver of poverty. And as Jeffrey Sachs from Columbia University has been saying for many years, one of the reasons these countries do have trouble getting out of this ruthless cycle of poverty is because an issue for them, for their normal citizens every day when they wake up is, am I going to get sick? Is my child going to get sick? And is my child going to die? To the point where it just becomes part of life. I read the newspaper every morning. For them, they're dealing with that consistency all the time. And I can't imagine the sort of effect that has on their life. So we're going to do some biology. There's some very, very basic biology about how malaria is transmitted. And I'm going to go into this simply because the question that we're raising is, okay, if climate change proceeds as we really do believe it will, and the Earth gets warmer, will we see that distribution change? And if I tell you that if we look at abiotic features which actually impact insect distribution, temperature is one of the major ones. If I am rearing mosquitoes or rearing Drosophila in the lab and I want to increase their life cycle, I can just simply turn up the thermostat. So it stands to reason, if you just sort of think about it from almost first principles, that, well, if the world gets warmer and there's more water around because mosquitoes need water to breed, well, yeah, maybe this will happen. Maybe that is going to happen. Maybe if somebody got up and said, my gosh, in 2050, we're going to have malaria all over the place. Maybe that makes sense. And I'm not going to tell you that that will or will not happen, but I want to illustrate that it's actually a very complex problem, and we need measurement. Okay? So, mosquitoes are not flying syringes. What happens when we're talking about the life cycle of the parasite called Plasmodium, which actually transmits malaria, is that it bites an infected person, and then this parasite, for a period of time, lives in the gut, the midgut of the female mosquito. It's only female mosquitoes bite. The reason for that is they want your blood to make their eggs, and that might sound unsavory, but that's reality. And then the parasite, then once it's in the gut of the mosquito, can complete part of its natural life cycle. And depending on the temperature, and if we're talking Central America, Northern South Africa, South Asia, Equatorial Africa, they can be quite warm. This will occur in 10 days or so. These will migrate as part of their normal developmental cycle into the salivary glands, and then if she's lived long enough, so we're now maybe 2 weeks after this event, if she's lived long enough and wants to make more eggs, she'll look for another hapless victim, bite them, take more blood, and transmit the pathogen into the bloodstream of the second person who's bitten, and the parasite can now complete the human part of its life cycle. So clearly, if this is going to happen, two things need to occur. First of all, this mosquito has to live long enough to bite a second person, and some people who are looking at insect control strategies have actually found cunning ways to make this shorter, but that's not the subject of this talk. And the second thing that has to happen is that clearly this person has to be infected, but if the life cycle of the parasite is going to be continued, this second person has to be non-infected. And so newly born infants, toddlers, are prime victims because they haven't been previously infected. So there's a number of parameters that need to be in place if we're going to have an infection, and some of those are independent of temperature, but some of those are. And what vector biologists, and when I start using the term vector, I'm talking about insects that vector disease. So I'm going to be probably saying that a mosquito is a vector of, in this case, Plasmodium. So I'm just going to sort of lapse into using the term vector. Because we're scientists, some people have come up with an equation to try and explain what the likelihood of transmission of these pathogens are. So I'm talking about malaria, but I could be talking about a virus, or I could be talking about a worm that's in mosquitoes. And these forces of transmission, basically they measure the capacity for spread and maintenance of an infectious agent in a host population. And one measure is something that's called the basic reproduction number. Now, I'm not going to show you the equation. It's not the point of the exercise. What I want to simply illustrate is that let's have a look at the parameters of the equation. And there are 9 parameters of the equation. Only 8 is shown here because one is just a natural logarithmic variant of one of these parameters. And what I've shown in blue are the ones which are dependent on the host. So if we're talking about malaria, that's us. But the rest of them, whether they're in black or red, are properties of the vector, the mosquito. And it's a very good rule of thumb in entomology that if you want to eliminate these diseases, you target the vector. And you can see here that all these variables are relating to the vector. Now, the ones in red are the ones that are directly affected by temperature. The ones in black are not. When you look at this, what I want you to think about is that if you're an entomologist or an epidemiologist and you're studying the transmission of malaria, of the Plasmodium in Africa or in South Asia or in Central America, how do you actually measure these? Because if this equation is of any use to us, then we have to have good measurements. We have to be able to plug values in. So we have to be able to measure the vector-host ratio. How many mosquitoes are there? How many naive people, in other words, uninfected people? What's the daily biting rate of those mosquitoes on infected hosts? What's the vector, the mosquito's daily survival rate? Now you think if that was you, or if that was me, sitting in that hut in Kenya or Tanzania, you can probably say, well, yeah, I can probably think of a way to measure whether that mosquito is going to survive today, and if I count enough mosquitoes, I'll figure it out. But I think you can see that it's probably going to be quite problematic to get those data. And in fact, even though this equation, by and large, is correct, the problem is getting good values for it. And I'm going to come back to that issue in just a moment. But before we get into the example I wish to use, we should also be aware of the history of malaria. So at the moment, there's no malaria in the United States. That's been the case for some time. But actually, naturally, before Europeans, malaria was right through the United States. And so here's some historical data from the CDC showing the distribution or malaria states of the Union in 1882. And it's a bit fuzzy, but you can actually see that most of the country is covered in malaria, and also the Central Valley. Here's the situation in 1912. Here's the situation in 1932. So now we're starting to understand mosquitoes transmit Plasmodium, and we start to take preventative measures, draining swamps, better housing. And here's the situation in 1943. And you can notice that it actually went backwards, maybe due to a heat spell, maybe due to the effects of the Great Depression and lack of public financing. And if we looked at the rest of the world, you would see that way back when there was malaria in Europe, and there was malaria through Australia and many other places. And our ingenuity in public education and public health has eradicated that from many parts of the world. So here, once again, it's some old CDC data. It's just basically showing you the mortality and morbidity rates of malaria in the United States. And if we look at 1920, we had about 350 cases per 100,000 and about 60 deaths. And obviously going down. And the big push was after the Second World War when, and then the development of insecticides. And that really did eliminate malaria from this country. You could say, well, you know, we've got rid of malaria in the United States. So even if the temperature increases over the next 50 years, it's not going to come back because you guys got rid of the mosquitoes. And the answer is that that's wrong, because the mosquitoes are still there. We got rid of the pathogens, but we did not get rid of the mosquitoes. So here's a species called Anopheles freeborni, which is a natural, inhabitant of this part of the country, of the western coast of the United States. It's a vector of, or it was a vector of malaria. It could be again. These are some recent isolations in this part of the world, and here's its potential distribution. And we could look at the whole world. We don't need to see what all these colours are. These are all different colours, show different species of Anopheles mosquitoes. All of these can transmit malaria. And so, Anopheles quadrimaculatus, which is one of the principal vectors in the central and eastern United States, is still there. And so if we lost complete control and somehow Plasmodium was reintroduced, potentially we could have this distribution finding itself again. Now I want to get into this very quick analysis, very briefly. And I want to talk about the work by Rogers and Randolph that was done 10 years ago and was asking the question in response to data that had been published and shown that, in fact, if one was looking at climate change and the effects on malaria distribution, this was going to really become quite extreme. And these authors were alarmed at that because they felt that those models, those predictions were inaccurate and were flawed in the sense that the authors of other studies, which were showing a dramatic expansion in the range of malaria in the world in response to climate change, they were using that equation. Now, there's nothing wrong with that equation that I didn't show you. But I showed you the parameters, and I said, how hard is it to measure these parameters? And the point of the exercise is that when you looked at those data that said, here's a potential rapid expansion of malaria through the world in response to climate change, a lot of those measurements were not made precisely, and lots of assumptions were made, and lots of values of 1.0 were plugged in, primarily through expediency. So what these authors did, Rogers and Randolph, were saying, well, we believe in that equation, but it's not appropriate to use it because we can't get good measurement. So rather, let's just look at climatic data. And what they did was that they went to 1,500 randomly selected sites where there was malaria in the world and basically measured temperature and temperature and moisture over the preceding 30 years. And they selected another 1,500 sites that were within 10 degrees of latitude of that initial site. So you had pairs of sites throughout the world. And they developed a model that says, well, can we then estimate, can we predict what the current, in 2000, distribution of malaria is? And on this top chart, or this top map, everything in green is where they predicted malaria would be in 2000. And that yellow hatch is the actual distribution of malaria. And if you just looked at Africa, you can actually see a pretty good correlation. Maybe not so great around here in southern Brazil. Central America, it's pretty good, and through South Asia and Southeast Asia, and then into the Fertile Crescent. That's a pretty good correlation. Not perfect, that's not bad. So they then took those data, they then took their model, their climatic model, and said, well, if we assume a 1% increase in the concentration of greenhouse gases, it's compounding 1% increase over the next 50 years, then what will the distribution of malaria look like in 2050? And the central panel shows that change. And you may have trouble seeing where these changes are, so what they then did is a simple subtraction, and in the bottom panel you can actually see in those regions that are coloured where malaria will be new and where it won't. And where it's hatched, you can just simply assume it'll still be there. It's just basically no change between 2000 and 2050. And so in these red areas down here, so this is the difference, okay? So if we're talking potential distribution of malaria in response to climate change, it's actually not that great. And this was in stark contrast to other models which said we'd have tremendous expansions. Now, I just want to go through this in just a little more detail. So in red here are areas that are presently suitable, present as in 2000, but becoming unsuitable by 2050 for the spread of malaria. So you could say that if you lived in the Horn of Africa, then maybe the incidence of malaria will go away. But that if you lived in these areas where there's green, presently unsuitable for malaria, but becoming suitable by 2050, you may have a problem. But from a global point of view, the net gain or loss is not that great in terms of human suffering. But if you live here, or if you live in southern Brazil, or you live in northern Iran, or the southern states of the steppes of Asia, or you live parts of China, or you happen to live anywhere between Cairns down through Brisbane into Coffs Harbour, you may disagree. I'm not saying this is right. I'm not saying the other one, which actually showed a more dramatic expansion, is right. I'm simply saying what's important here is the value of measurement. And that we have equations which can actually forecast the ability for these pathogens to move around, but how we actually measure information is important when we determine what the consequences of climate change can be on the distribution of pathogens transmitted by insects, and malaria is just one example. We can get things wrong. No one in 1998 would have believed that a single, or maybe two or three, but maybe just a single infected mosquito coming into New York City from an undisclosed location would within 4 years lead to a new disease, West Nile virus, being in every state of the United States. But that did happen, and the United States is arguably the most sophisticated, advanced country in the world. It has magnificent abatement districts with dedicated staff, has a great public health system, has great communications, great public awareness. We all knew what was going on, and we could do nothing to stop it. And so this just simply shows the spread by state from the initial introduction in New York right through to more or less the current day. Now, This did not have a high mortality. It certainly had a high mortality in horses of about 33%, and the mortality rate in humans was about 3 to 5%. And unfortunately, for those who got West Nile, there was about 30 or 50% of them had presented with serious neuropathologies. So for those people, it was a serious issue. But this is not malaria in terms of its economic or public health significance. But the point of the exercise is we can talk about models and we can talk about reality. And this was independent of climate change. The second example I want to very briefly go through is not as elegantly or elaborately worked out. It's by a scientist called Ken Linthicum, who works at the USDA in Gainesville, Florida. And we had a meeting here 2 years ago where he actually presented some data, modelling data, based on the El Nino event of 2006. So here's what I understand to be the El Nino event towards the equatorial part of the Eastern Pacific. And in previous years around the world, there'd been a correlation between El Ninos and the prevalence of mosquito, vector-borne disease, some months after in various parts of the world. And so when this 2006 event occurred, there were predictions of the following: that there might be an increase in plague in this part of the country, there might be an increase in malaria in this part of South America, in dengue in Brazil, in malaria and Rift Valley fever, which is an arbovirus transmitted by mosquitoes in Eastern Africa, specifically in the Eastern Horn, in cholera in India, which is not transmitted by insects, and what's not very hard to read here is dengue in Indonesia and related countries. And so the question is, did any of those happen? And the answer to that is, this didn't happen, this didn't happen, this probably happened. There was a 20% increase in dengue transmission in Brazil. This didn't happen, this most certainly happened. And the Indonesian government in 2007 was reporting almost catastrophic levels of dengue transmission. And this actually also happened more to do with Rift Valley fever than it did with malaria. So 3 out of 5 is not bad. You can actually do forecasting based on known climatic change events. And I actually want to point out something very interesting about this case of dengue. Because dengue is a virus, it's transmitted by mosquitoes, and I told you that the larval life stage of mosquito requires water. So if we think of climate change and we think of increased temperature, and maybe depending on where we are, we may have an increased rainfall and increased humidity, you might be sitting there and saying, well, obviously this part of the world's hot anyhow, it's on the equator, and it probably got a little hotter, maybe in response to this El Nino effect, and it probably rained more. But in fact, that's not true. The monsoons failed, and there was drought. There was drought here. Mosquitoes need water. So how can it possibly be that drought leads to an increase in the prevalence of dengue? And the answer is brilliant. Because here, when there's drought, as we do, people gather water. Well, here we preserve water. But if there's no running water, you have to get your water in buckets or containers. And so if there's drought, in these households, in a developing country, you have more standing water being transported in and around homes in open containers for people to drink from. And that's a perfect breeding site for mosquitoes, and it's extremely close then to the people they bite and transmit diseases to, in towns or cities like Jakarta, and even Singapore, which is an advanced city in that part of the world. So if we're talking about that equation, here are more parameters that we don't plug into that equation, dealing with cultural values. How do we actually treat water? Can we afford closed containers or not? It's illustrating how complicated this can be, or forecasting can be. Very briefly, for the last 5 or 10 minutes, I simply want to talk about how we can approach this issue with genetics. Many of those parameters that I spoke about in that equation have their foundation in genetic variation. If we went back to the model of the mosquito, transmitting the plasmodium. And remember, the plasmodium is living in the female mosquito for 10 days and has to transverse from her midgut up into her salivary glands. I mean, there are genetic interactions there. And we're starting to understand what they are. But there's another part of this, and that is, well, if all insects, whether they're beneficials or they're pests, are going to be impacted by climate change, and let's just talk about increasing temperature or increasing desiccation or a reduction in desiccation, how are insects going to be able to respond to that? Let's just think about the beneficials, things that we never see. What's it going to be doing to biodiversity in the tropics of insects in Amazonia or in Central America or in Africa? What's it going to be doing to the diversity of insects in temperate regions? And many authors, primarily the ones I've shown here, have developed this climatic variability hypothesis, which simply states that insect distribution is controlled in part by environmental variables, and let's just talk about temperature. And then the observation's been made that species distributions become narrower towards the tropics. And the hypothesis proposes that species with higher levels of physiological tolerance to changes in climate should have more success in moving into higher latitudes, north and south of the equator, depending on where they are, and that species do not have these levels of tolerance tend to be confined to the tropics, and that this has all been underpinned by genetic variability. The bottom line is that the expectation, if we were going to test this hypothesis, is that insect species that live in the tropics will be less able to deal with changes in climate than those in temperate regions. And if we're worried about biodiversity of insect species in the tropics, butterflies, whatever, then that becomes an issue. Well, can this be tested? And the answer is that it can. And so work by Ari Hoffman and his colleagues at the University of Melbourne that was published just this year took advantage of 30 species of Drosophila that he had available to him, and he simply stressed them with exposure to cold, although we could do heat if you wanted, and desiccation. So desiccation is drying out. It's the reverse of increased humidity. And the question he simply was asking in these trials was, did Drosophila species that had a widespread distribution have greater resistance to being dried out or placed in the cold than tropical species? And you can see by those two coloured circles that the climate variability hypothesis was proven, in that species, insect species from temperate regions, which the hypothesis says should be better able to adapt through their increased genetic variability, should be more resistant to these climatic stresses than ones from the tropics. And even though this experiment obviously hasn't been done for a whole range of species, these experiments with Drosophilids are deeply informative of what we may be confronting if the tropics warm up, in terms of the impact that that may have on insect biodiversity. Now, Hoffman and his colleagues didn't identify those genes that were involved in providing those Drosophilids with resistance to desiccation or resistance to cold. And you could say, well, we may never find out what those genes are. But in fact, we now are getting the technologies that enable us to do that. So these scientists from the University of Arizona work on cactophilic Drosophilids, which actually live, breed and eat on cacti. There's a particular species called Drosophila mohovensis, which not surprisingly lives in the Mojave Desert, and its genome's been sequenced, along with 11 other Drosophilid species. These days it's getting very easy to sequence whole genomes of many organisms. And I don't want to go into the techniques here, simply to say that the vast majority of them are automated and they don't require a lot of hands-on work. But the question is, could these authors identify genes from Drosophila mohovensis that were actually being transcribed or read more often in response to stress? And they were able to find that there were 4 genes involved in basic metabolism, I won't list them, that were involved in this. And clearly, they could be candidates for the type of study that Hoffman and his colleagues undertook on those Drosophila species. This gets to my final point, because I'm going to go back to those three boxes I showed you. That we had one box up the top, which was climatic information, a box down here, which was dynamics of interactions between pathogens and their hosts, and one over here, which actually was talking about insect genomics. And the theme of this talk has been about measurement, and how accurate measurement is absolutely required if we're going to make sense of predictions of distributions of change of insect species in response to climate change. And as I pointed out, the climatologists are way ahead of us. Well, now with high throughput whole genome sequencing, finally, in the history of the planet, the technology catches up to the diversity of insects. And it's becoming very cheap, very easy to get whole genome sequences of entire organisms. So you may recall that some 10 years ago, after many years' work, we got the first sequence of a human. Now we're talking about sequences of 1,000 humans. And very soon there'll be sequences of 5,000 humans. If you apply that technology to other organisms, especially insects, which typically have smaller genomes from humans, what we now are heading towards is that population genetics at the genome-wide level can be done for any insect species which we're asking questions about. Genes involved in an insect's response to climate change can be identified and their frequency in a population determined. And the impact of genotype on the force of transmission of pathogens, whether we're talking about a mosquito transmitting malaria, a mosquito transmitting a virus, or an aphid transmitting the virus to an agricultural crop, can actually be determined. And this formula down the bottom, which is a very simplistic one, is one of the cornerstones of genetics. Genotype plus environment equals phenotype. I'm a geneticist. I work on this. Ecologists, environmental scientists work on this, but that produces the phenotype. And the phenotype is the presentation of the organism. What I look like, my behavior, or in the case of an insect, what its ability to withstand or tolerate climate change may be, or its ability to transmit a pathogen, or its inability to do so. There's a genetic basis to that. And so for the first time, we have the ability to start measuring these things. And so finally, in work that came from colleagues here at Riverside and Peter Ahrensberger in my own lab, and I'm not going to go into this in any detail, I just want to illustrate that those three blobs up there, doesn't matter what those numbers are, just look at those three big blobs. They represent total numbers of genes in 3 species of mosquitoes: Culex, which, if you like, is a vector of West Nile; Anopheles, which is a vector of Plasmodium and malaria; and Aedes aegypti, which is a vector of many things, but let's just talk about dengue, okay? These are from that type of analysis, that whole genome analysis and expression analysis. These are the subsets of all genes involved in these mosquitoes in resistance or immunity to all these pathogens. And in the middle of this Venn diagram is just those genes that are common to all 3 species, regardless of which pathogen they've been infected with. And these ones here are in common between Culex and Anopheles, regardless of whether this has been infected with Plasmodium and these have been infected with worms, bacteria, or a virus. There's a huge amount of genetic information in here which I'm not showing you. Reams and reams of data. But within a very short period of time, these scientists were able to unravel all, or the core material required in the genetic interaction of these mosquitoes with the pathogens they transmit. Now let's get, why am I telling you this? Well, this is a revolution, and it increases our ability to measure things. But recall I was talking about that equation showing you the force of transmission of a pathogen. And remember when we went through the life cycle of the mosquito, when the Plasmodium entered that mosquito's midgut, and over the course of 10 days to 2 weeks, depending on the temperature, migrated to her salivary glands, that was temperature dependent. If we discover the genes in the mosquito or in the aphid or whatever pest you're talking about, that are implicated in that progression of disease, and then understand the frequencies of variants of them in the population and how different variants of those genes may affect the ability to transmit disease in response to increasing temperature, then suddenly these black boxes start to vanish. And we're not here yet. As I said, through the fantastic work of climatologists, this black box, the climate box, is now an open, clear box. And all those graphs I showed you at the start and Dr. Kennedy showed you 6 weeks ago are evidence of that. These boxes are still pretty dark. I've just shown them in grey. This is actually where we're headed. These are the genotypes, the G in that G plus E equals P equation. Any species of insect, population biology, we are now, or we're starting to be able to approach. And if we start talking about what the dynamics of transmission within the insect are and how these may respond to changes in temperature, that black box starts to become a grey box and soon becomes a white box. And so they produce the phenotype which leads to informed predictions, not guesstimations, not rule of thumb, not heurisms, not guessing, not noise. Reality, hard data. And that leads to appropriate proactive decisions. Because in the end, we don't want to be the cork bobbing in the ocean. We go back to that original graph showing the increase in carbon dioxide concentration in the atmosphere, and I said, we take ownership of it because all of us in this room are part of that. We are directly or indirectly responsible, and it's our choice what we do about it.